134. Plant growth and mineral nutrition. 1. Do you think that the growth is property of living beings only? 2. Is there any difference between plant growth and animal growth? Can you recall? 7. 7.1 Plant growth. Growth is one of the characteristic features of living organisms. Growth as a phenomenon has two aspects, viz. Quantitative and qualitative. Quantitative aspect speaks for an increase in the length, breadth, size, volume, body mass or dry weight and number of cells. Growth as a quantitative change is a final end product of successful metabolism. Qualitative aspect talks about the change in the nature of growth, where development is an ordered change or progress while differentiation leads to higher and more complex state. Growth thus can be defined as permanent, irreversible increase in the bulk of an organism, accompanied by the change of form. In multicellular, vascular plants, growth is indeterminate and occurs throughout the life indefinitely. It is restricted to some specific regions called meristems, which are the regions where new cells are constantly and continuously produced. Meristems are of three types based on location, viz. Apical, intercalary, and lateral. 7.2 Phases of growth. The cells in the meristem divide, enlarge and get differentiated. Corresponding to these three stages, there are three phases of growth. A phase of cell division formation. Cells of meristem are thin-walled, non-vacuolated, having prominent nucleus and granular cytoplasm. Meristematic, particularly cambial, cell undergoes mitotic division to form two new cells. One cell remains meristematic and the other cell undergoes enlargement and differentiation. In this phase, rate of growth occurs at a slower pace. Lag phase. B phase of cell enlargement elongation. The newly formed cell becomes vacuolated, osmotically active and turgid due to absorption of water. The turgidity results in the enlargement of cell, both lengthwise and breadthwise. In this phase, new wall materials and other materials are synthesized to cope up with the enlargement. The growth rate in this phase occurs at an accelerated pace. Exponential or log phase. Apical meristem. In vascular plants, growth is restricted to the apices of root and shoot. It is responsible for growth in length, height and the differentiation or cell types. It contributes to the primary growth. Intercalary meristem. It is located at the node or at the base of internode of stem. It is primarily responsible for increasing length of internodes and also for formation of leaf primordia and lateral buds. Lateral meristem. It is located laterally along the axis of dicotyledons and gymnosperms. It is located as strip in the vascular bundles of stem of dicots. It is called vascular cambium. It is responsible for increase in the girth of the stem due to addition of secondary vascular tissues. Do you know? Root and stem show indefinite or indeterminate growth. However, in organs like leaves, flowers and fruits, growth is determinate, i.e. they grow up to certain genetically destined size. In unicellular plants, growth is uniform and determinate. Do you know? 135. Fig. 7.1 Phases of growth in root. Position of radicle at the beginning, A, and at the end, B. Plumal. Zone of mature cells. Zone of cell differentiation. Zone of cell elongation. Zone of cell formation. A. B. C phase of cell maturation differentiation. The enlarged cell now becomes specialized to perform specific function and attains maturity, both morphological and physiological. In this phase, rate of growth slows down and comes to a steady state, stationary phase. Growth in plants can be measured in terms of 1. Increase in the number of cells produced, e.g. single match root apical mesostem can give rise to more than 17,500 new cells are. 2. Increase in surface area of the leaf, e.g. growth of dorsiventral leaf. 3. Increase in length, e.g. growth of pollen tube. 4. Increase in volume of a fruit, e.g. in watermelon flower, the ovary after fertilization increases in its size volume by up to 350,000 times. 5. Increase in girth of shoot. 6. Increase in dry weight of organ. Various methods for the measurement of linear growth of stem and radicle are as follows. 1. Direct method. It can easily be measured with the help of ordinary measuring scale. It is a simple method. 2. Horizontal microscope. It is used to measure growth in fields. 3. Oxanometer. This equipment is used for precise measurement of linear growth of shoot. There are two types of an oxanometers, viz. A. Arc oxanometer, B. Pfeffer's oxanometer. 4. Crescograph. It records primary growth very accurately. It magnifies growth up to 10,000 times, giving information of growth per second. It is developed by Sir J. C. Bose. 7.3 Conditions for growth. The different environmental and physiological conditions necessary for the growth include water, supply of nutrients, temperature, oxygen, carbon-nitrogen ratio, gravitational force, light and growth hormones. The chief conditions are explained below. Water is the essential component of protoplasm and maintains turgidity of the cell. It acts as aqueous medium for biochemical reactions. Microelements and macroelements are nutrients required for the proper growth of the plant. Optimum temperature ranges between 25 to 350 C. Oxygen is essential for respiration and the release of energy. Light is very much essential for germination of seed and photosynthesis. Gravitational force decides the direction of growth of the shoot and root. 7.4 Growth rate and types of growth. Growth rate, it is the increased growth per unit time. It is also called efficiency index. Rate of growth can be measured by an increase in the size and area of different plant organs like leaf, flower and fruits. The ratio of change in the cell number, dn, over the time interval, dt, is called absolute growth rate, agr. Alternatively, it is the measurement and comparison of total growth per unit time. 136. Ag is equal to dn dt. The Ag, when divided by total number of cells present in the medium, gives relative growth ratio, RGR. Alternatively, RGR refers to the growth of a particular system per unit time, expressed on a common basis, or it is the ratio of growth in the given time initial growth. RGR is equal to Ag. Ag and RGR are useful in describing the dynamics of cell growth in culture. Types of growth. There are two types of growth, viz. Arithmetic growth and geometric growth. Arithmetic growth. Here, rate of the growth is constant and an increase in the growth occurs in arithmetic progression, i.e. 2, 4, 6, 8 cms etc. In this type of growth, the rate of growth is constant. After mitosis, one of the daughter cell continues to divide and the other cell takes part in the differentiation and maturation, e.g. elongation of root at a constant rate, best explains arithmetic growth. Linear curve is obtained when growth rate is plotted against the time. Arithmetic growth is expressed mathematically by an equation. It is expressed as lieutenant is equal to low plus RT, where lieutenant is equal to length at time, T, low is equal to length at time, 0, R is equal to growth rate, T is equal to time of growth. When graph of length, L, is plotted against the time, T, a linear curve is obtained as indicated in the diagram. B. Geometric growth. Cell divides mitotically into two. Here, both the daughter cells continue to divide and redivide repeatedly. Such growth is called geometric growth. Here, growth rate is slow. W1 is equal to VO. W1 is equal to final size, VO is equal to initial size, R is equal to growth rate, T is equal to time of growth, E is equal to base of natural logarithm. Graph 7.2, constant linear growth. Height of the plant, L. Time, T. A. B. Fig. 7.3, growth rate. A. Arithmetic. B. Geometric. Zygote divided. Geometric phase, all cells divide. Arithmetic phase, these cells divide, these cells do not divide. Is equal to cells capable of division is equal to cells that have lost capacity to divide. Diagrammatic representation of A. Arithmetic, B. Geometric growth and C. Stages during embryo development showing geometric and arithmetic phases. C. Initially but later on there is a rapid growth at exponential rate. Geometric growth can be expressed mathematically by an equation. 137. Fig. 7.4, growth rate comparison. A1. A. 10 cm2. B1. 55 cm2. 5 cm2. B 50 cm2. The total time, period, required for all phases to occur, is called grand period of growth. 7.6 Differentiation, dedifferentiation, redifferentiation, adifferentiation. It is maturation of cells derived from apical meristem of root and shoot. Permanent change in structure and function of cells leading to maturation is called differentiation. During cell differentiation, cell undergoes few to major anatomical and physiological changes, e.g. parenchyma in hydrophytes develops large schizogenous interspaces for mechanical support, buoyancy and aeration. The maturation is at the cost of capacity to divide and redivide. B. Dedifferentiation, the living differentiated cell which has lost the capacity to divide, may regain the same as per the need and divide. Thus, permanent, mature, cell undergoes dedifferentiation and becomes meristematic, e.g. interfascicular. We can also observe quantitative comparison between the growth of living system in two ways. Measurement and comparisons of total growth per unit time is called the absolute growth rate, AG, whereas the growth of the given system per unit time expressed on a common basis per unit initial parameter is called the relative growth rate, RGR. In the above example, two leaves, A and B, are of different sizes but show same absolute increase in area in a given time. Both leaves grow and increase their area by 5 cm2 to produce A and B leaves. A leaf of 5 cm2 in size grows 5 cm2 day, then its RGR would be 100%. If the leaf is 50 cm2 in size and the growth rate day is 5 cm2, then its RGR would become 10%. 7.5 Growth curve. It is a graphic representation of the total growth against time. There are three types of curves, viz. Linear, exponential and sigmoid. Arithmetic growth curve is linear while geometric growth curve is exponential. Corresponding to three distinct phases of growth, growth rates differ. In lag phase, growth rate is slow. In exponential, log, phase, growth rate is faster and reaches its maximum. In stationary phase, growth rate gradually slows down. When a graph of rate of growth against time is plotted for three phases of growth, a S-shaped i.e. sigmoid curve is obtained. Graph 7.5, exponential growth curve. J-shaped curve. Population size. Year. Graph 7.6, sigmoid growth curve. Size weight of the organ exponential phase. Lag phase. Stationary phase. Time. 138. 7.8 Plasticity. It is the capacity of being molded, formed or modeled. It is the ability of plants to form different kinds of structures, i.e. to change, in response to different environmental, external, or internal stimuli, in various phases of life. In many plants, juvenile stage and mature stage show different forms of leaves in the same plant, e.g. heterophyly in cotton, coriander, larkspur, delphinium. The environmental heterophyly is shown by Ranunculus flabellaceus, buttercup. The intrinsic plasticity is found in coriander and cotton. Heterophyly is exhibited in the same plant in different growth phases or under different environmental conditions. Cambium and cork cambium are formed from parenchyma cells between vascular bundles and innermost layer of cortex, respectively. See redifferentiation, The cells produced by dedifferentiation once again lose the capacity to divide and mature to perform specific function. This is called redifferentiation, e.g. secondary xylem and secondary phloem are formed from dedifferentiated cambium present in the vascular bundle. 7.7 Development. It refers to the ordered or progressive changes in shape, form and degree of complexity. It includes all the changes occurring in sequence from the germination of seed up to the senescence or death during life cycle of plants. Thus development includes growth, morphogenesis, maturation and senescence. Fig. 7.7 Flow chart of development. Seed germination. Meristem. Cell division. Plasmatic growth. Cell elongation. Cell maturation. Mature cell. Senescence. Death. Fig. 7.8 Heterophylla. 1, 2, 3. 4. Juvenile adult terrestrial habitat. Water habitat. 7.9 Growth hormones. The term, hormone, was coined first by Starling, 1906, in Animal Physiology. The internal factors that influence growth are called growth hormones or growth regulators as they inhibit, promote or modify the growth. Growth promoters are auxins, gibberellins, Ga, and cytokinins, Ck. Growth inhibitors in plants are ethylene and abscisic acid, ABA. All phytohormones are growth regulators. 1. Is there any relation between phases of growth and regions of growth curve? 2. Which plant organ does show both arithmetic and geometric growth? 3. Differences between arithmetic and geometric growth. Can you tell? 139. According to Thiemaner and Pincus, 1948, plant hormones are organic substances produced naturally in higher plants affecting growth or other physiological functions at a site remote from its place of production and active in very minute, optimum, amount. Hormones are transported through phloem parenchyma, Phillips 1971. Auxins, auxion is equal to grow FW Wendt in 1931, used this term first. Auxine was isolated from urine of a person suffering from pellagra, Coble and H. Smith 1931. In plants, it is synthesized in growing tips or meristematic regions of plants from where it is transported to other plant parts. The most common and important natural auxine is indole 3-acetic acid, IA. Tryptophan is the primary precursor of IA in plants. It is the first hormone to be discovered in plants and is primarily responsible for cell elongation. It shows polar transport, basipetal transport in stem. Now synthetic auxins like IBA, indole butyric acid, Na, naphthalene acetic acid, 2,4-D, dichlorophenoxyacetic acid, etc. are used. Physiological effects and applications of auxine. The primary effect is cell enlargement. In most of the higher plants, growing apical bud inhibits the growth of lateral buds. This is called as apical dominance. Auxine stimulates growth of stem and root. Auxine induces multiplication of cells, hence used in tissue culture experiments to produce callus. It stimulates formation of lateral and adventitious roots. These are marketed as synthetic herbicides, e.g. 2,4-D, 2,4-dichlorophenoxyacetic acid. It kills dicot weeds without affecting monocot crop plants. The seedless fruits like orange, lemon, grapes, banana etc. are produced by application of auxine, i.e. induced parthenocarpy. Auxins promote cell division in cambium and also cause early differentiation of xylem and phloem. It. Know the scientist. The auxine is the first hormone to be discovered in plants. Discovery of auxins dates back to 19th century when Charles Darwin, 1886, was studying tropism in plants. He exposed canary grass coleoptile to unilateral light. He concluded that a growth stimulus is developed in the coleoptile tip and transmitted downwards to the growth zone. This has caused bending of the tip towards light. The Danish plant physiologist Boysen Jensen, 1910, cut off the colpothyle and inserted thin plate of gelatin between the tip and the cut stump. He observed that coleoptile tip still bends towards unilateral light. Paul, 1919, cut off the tip of colpothyle and replaced it asymmetrically on the cut coleoptile stump. He observed that the colpothyle tip bent away from the side-bearing tip even in dark. F.W. Wendt, 1928, successfully isolated natural auxine from Avena coleoptile tips. He cut off the tip and placed them on small agar blocks. Then after certain period of time placed the agar blocks asymmetrically on cut coleoptile stump that caused bending. He demonstrated the presence of substance which could diffuse into agar blocks. Wendt named this substance as auxine. Coleoptile bends tip removed coleoptile tips on agar. Top of coleoptile removed, no bending, gelatin inserted tip placed on one side tips removed, agar cut into blocks. Light-proof cap on tip, no bending, bends towards light bending in absence of light agar block placed on one side and bending occurs Darwin Boysen Jensen Paul Wendt. 140. B. Gibberellins, it is another growth-promoting hormone and is abundant in root tip and developing seeds. It shows non-polar transport through vascular tissue. Gibberellins were first isolated from the fungus Gibberella fujikuroi by a Japanese scientist Kurosawa, 1926. He observed that when rice plant was infected by fungus Gibberella fujikuroi, it shows extensive stem elongation called Bakane disease. The crystalline form of gibberellins were isolated by Yabuta and Sumiki, 1938, from the fungus culture. They named it as gibberellin. It is synthesized in young leaves, seeds, roots and stem tips. These are synthesized from mevalonic acid. More than 150 chemical types are known so far. Ga3 is equal to gibberellic acid, is most common and biologically active form. Chemically it contains a gibbian ring acyclic diterpene with 4 isoprene units. Physiological effects and application of gibberellins, dormancy of bud can be broken by Gibberellin treatment. It can promote seed germination in cereals like barley and wheat by synthesizing hydrolysing enzyme amylase to produce sugar. The most striking effect of it is the elongation of stem where internodes increase in length and converting genotypic dwarf to phenotypic tall plant. It also promotes bolting eye. Elongation of internodes just prior to flowering in plants those with rosette habit e.g. beet, cabbage. It causes parthenocarpy in tomato, apple and pear, and flowering in long day plants. It is used to increase the fruit size and bunch length of grapes. When gibberellins are applied on genetically dwarf plants like match, the stem rapidly elongates and acquires the height of normal tall varieties of match. Application of gibberellins overcomes the requirement of vernalization. Usually, it inhibits growth of root, delays senescence and prevents abscission. It also breaks dormancy of seed and hastens germination. Application of gibberellin causes production of male flowers on female plant. See cytokinin. It is another growth hormone that promotes cytokinins during cell division. Latham coined the term cytokinin. The first cytokinin was discovered by Skoog and Miller, 1954, during investigation of nutritional requirements of callus tissue culture of Nicotiana tabacum, tobacco. They observed that the callus proliferated when the nutrient medium was supplemented with coconut milk and degraded sample of DNA, obtained from herring sperm. They named it as kinetin. Chemically kinins are 6-furfuryl-aminopurine. First natural cytokinin was obtained from unripe match grains by Latham et al. It is known as zeating. 6-benzyladenine is a synthetic cytokinin hormone. 7 different types of cytokinins are recorded from plants. Natural cytokinins are also reported from plants like banana flowers, apple and tomato fruits, coconut milk, etc. Physiological effects and applications of cytokinin. Besides cell division, it also promotes cell enlargement. High cytokinin promotes shooting. Promotes root elongation in low concentration and shooting at higher concentration. It also hastens early rooting in propagation by cutting. Foliar spray of Na and 2,4-D induces flowering in lychee and pineapple. Likewise, it prevents premature fruit drop in apples, pear and oranges, and also prevents formation of abscission layer. Auxins play a role in elongation of cell. It is known to increase rate of respiration. Auxins break dormancy in seed and promote quick germination. Agent Orange, mixture of 2-phenoxy herbicides in ester form Vis2, 4-D and 2,4,5-T, dioxin, is known as Agent Orange used in Vietnam War for defoliation of forests. Do you know? 141. A low ratio of cytokinin to auxin induces root development but a high ratio causes buds and shoot to develop. Cytokinin and auxin ratio and their interactions controls morphogenic differentiation. It promotes the growth of lateral buds and controls apical dominance by cell division. It delays the senescence or aging and abscission processes in plant organs. This was reported by Richmond and Lang, 1957. Formation of interfascicular cambium and expansion of cells are other functions. It also breaks dormancy and promotes the germination of seeds. Cytokinin reverses apical dominance effect. It induces RNA synthesis and formation of interfascicular cambium. D-ethylene, it is the only gaseous growth regulator. Denny, 1924, reported ethylene is effective in fruit ripening. Gunn, 1934, established that plants naturally synthesize ethylene. Crocker, 1930, proposed that ethylene is the plant hormone responsible for fruit ripening. It is a simple gaseous hydrocarbon with essential role in the fruit ripening. The most widely used compound as a source of ethylene is ethepone. It is synthesized in roots, shoot, apical meristem, ripening fruits, etc. E-abscisic acid, it is a natural growth inhibiting hormone. Carnes and Adicott, 1961-65, observed that the shading of cotton balls was due to a chemical substance abscissine I and 2. Waring, 1963, isolated a substance from buds of acid that can induce bud dormancy and named it dormin. These two identical chemical substances were given the common name abscisic acid. It is synthesized in leaves, fruits, roots, seeds, etc. Chemically, it is a 15-carbon sesquiterpenoid and is synthesized from mevalonic acid. Physiological effects and application of ABA. It promotes abscission of leaves and induces dormancy in many plants. It controls the dormancy in buds and seeds by inhibiting growth processes. It accelerates the senescence of leaves, flowers and fruits. It inhibits and delays cell division and cell elongation and suppresses cambium activity by inhibiting mitosis in vascular cambium. ABA could cause efflux of K+ ions from the guard cells and result in closure of stomata. So, it is known as an antitransparent. It acts as a stress hormone by inducing the plant to bear the adverse environmental conditions. It inhibits flowering in long-day plants but stimulates flowering in short-day plants. Physiological effects and application of ethylene. It promotes ripening of fruits like bananas, apples and mangoes. It stimulates initiation of lateral roots in plants and breaks the dormancy of bud and seed. It accelerates the abscission activity in leaves, flowers and fruits by forming of abscission layer. Ethylene inhibits the growth of lateral buds and causes apical dominance and retards flowering. It is associated with ethret. Ethepone is a 2-chloroethyl phosphoric acid, which releases ethylene after dissolving in water. Do you know? The enhancement of process of senescence of plants' organs. It inhibits flowering in most of the plants except pineapple. It causes epinasty, drooping, of leaves and flowers. It increases activity of chlorophyllase enzyme causing degreening effect in banana and citrus fruits. Degreening, it is the process of decomposition of green pigment in fruits usually by applying ethylene. This method is called trickle degreening. Collect more information about degreening. Internet, my friend. 142. 7.10 Photoperiodism. Higher plants reproduce sexually by producing special structures called flowers. Plants exhibit transition from vegetative growth to reproductive growth during which flowers are produced. Like vegetative growth, reproductive growth is also influenced by several environmental and nutritional factors. Among the environmental factors, light and temperature exert profound influence on flowering. The influence of duration of light is known as photoperiodism and that of temperature is vernalization. Light as an environmental factor influences germination of seed, vegetative growth, photosynthesis, etc. Light as a factor has three aspects viz. Quality, intensity and duration of light. It is the duration of light that has profound effect on flowering in higher plants. The term photoperiodism was used by Garner and Allard, 1920. They were studying the flowering behavior in plants, soybean and Maryland marmot variety of tobacco. They found that soybean plant flowers during late summer and tobacco variety during winter, irrespective of their germination and growing season. They studied effects of different temperatures, nutrition, soil moisture, etc. in respect of flowering. None of these were found to regulate flowering. However, experimentally they found that the exposure to specific duration of light, i.e. photoperiod, had profound influence on flowering. They examined the effect of day length on flowering by using artificial illumination. They concluded that the relative length of the day was most crucial in the growth and development of flowers, to which they coined the term photoperiodism. Based on the photoperiodic response, plants were classified in three categories viz. Short day plants, SDP, long day plants, LDP, and day neutral plants, DNP. Organs for reception of photoperiodic stimulus. Leaf is the chief organ for receiving the photoperiodic stimulus as demonstrated by Knopf, 1934. Defoliated plants will not flower even if the plants are exposed to proper duration of light. Photoperiodic stimulus. It is a chemical stimulus transported through phloem and is called phloragen, which is hormonal in nature. Photochemical receptors in the leaves are the bilirubin pigments, located in the cell membrane. These are called phytochromes. Blue wavelengths of light influences flowering. Do you know? A short day plants, SDP. These plants usually flower during winter and late summer when day length is shorter than the critical photoperiod. Critical photoperiod is that length of photoperiod above or below which flowering occurs. These are called bioassays for auxins, Avena-Kvecher test, gibberellins, alpha amylase bioassay, cytokinins, chlorophyll retention test, ethylene, triple P test, ABA, inhibition of alpha amylase test. Do you know? Collect information of synthetic plant hormones. Internet, my friend. 1. Why is auxin called a growth regulator? 2. Effect of gibberellin application on apple. 3. How can we overcome apical dominance? 4. Which is standard bioassay method for auxins? 5. ABA is called as stress hormone. Why? Can you tell? 143. Long night plants because they require long uninterrupted dark period night for flowering. If dark period is interrupted even by a flash of light, SDP will not flower. Some of the short day plants are dahlia, aster, tobacco, chrysanthemum, soybean, glycine max, cockleba, xanthium, etc. B. Long day plants, LDP. Plants that flower usually during summer are called long day plants. They require longer duration of light than the critical photoperiod for flowering. They are called short night plants as they require short dark period. When long dark period is interrupted by a brief flash of light, LD plants can flower e.g. radish, sugar beet, cabbage, spinach, wheat, poppy, etc. Seed neutral plants, DNP. These plants flower throughout the year round, independent of duration of light, photoperiod. They do not require specific photoperiod to flower. Therefore, they are called day neutral plants, e.g. cucumber, tomato, cotton, sunflower, madge, balsam, etc. Phytochrome, Hendricks and Bortwick, 1952, observed that flowering in SD plants is inhibited if dark period is interrupted even by a flash of red light of 660 nm. If it is immediately followed by far red light, 730 nm, then SD plants will flower. This observation led them to conclude that some pigment system in plant receives the photoperiodic stimulus. These pigment proteins are called phytochromes. The leaves possess light-receiving proteinous pigment called phytochrome that induces flowering. It exists in two interconvertible forms viz. Red, PR, and far red, PFR. When PFR absorbs far red light, it is converted into PR and vice versa. These are located in the cell membrane of green cells. 660 nm, red, PR PFR 730 nm, far red, darkness. During daytime, PFR accumulates in the plants. It inhibits flowering in SDP but initiates flowering in LDP. During dark period PFR changes into PR, which stimulates promotes flowering in SDP and inhibits flowering in LDP. Control of morphogenesis by light and phytochrome is called photomorphogenesis. Always remember. 7.11 Vernalization, Yarovization, temperature as environmental factor influences several physiological processes including reproduction. Temperature as a factor has three cardinal points viz. Minimum. Fig. 7.9, photoperiodism. A requires a relatively long uninterrupted darkness. B requires a relatively small duration of darkness, no darkness at all. Light critical dark period. Flash of light darkness. Flash of light. Male chairs proposed that chilling treatment may induce formation of hypothetical floral hormone, vernalin. Internet my friend.